Today we are going to perform disc diffusion test by Kirby Bauer's disc diffusion. Here we are leveling the tubes for the appropriate samples. After leveling the tube, we will prepare inoculum. 3 to 5 fresh colonies of a similar morphology are touched to make a direct suspension in sterile normal saline. And after that, the turbidity of that saline is adjusted to 0.5 McFarlane. We can use various methods to check the turbidity. Here we are using photometric device commercially available as DensiCheck. Or we can visually compare the bacterial suspension with that of corresponding McFarlane turbidity standard tube by holding both the tubes against a card with white background and contrasting black lines known as Vikram card. After inoculation preparation, you can see the Mueller-Hinton Agar plates are labeled with date and time along with sample number. Also, you can see the name of antibiotics are written over the plate beforehand. Next, we will inoculate the test plate. A sterile cotton swab is dipped in the inoculum broth, rotated several times and pressed firmly on the inside wall of the above fluid level to remove the excess fluid. The swab here is inoculated on the entire plate 3 times with 60-degree rotation and finally the rim to ensure an even distribution of inoculum. While performing the Kirby Bauer disc diffusion, we should always remember the 15-15-15 rule, that is inoculum preparation, that is turbidity adjusted within 15 minutes, lawn culture of the inoculum on MHA plate, that should happen within 15 minutes, and applying the disc on MHA plates, that should occur in another 15 minutes, and next we should incubate the MHA plate. Within the 15 minutes of lawn culture, a set of antimicrobial discs are placed onto the surface of inoculated MHA plate either manually or here we are using the sterile forceps to place the disc. After being placed, each disc is pressed down with the forceps to ensure complete contact with the agar surface. We should ensure the maximum number of discs per plate on a 100 plate for non-fastidious organisms should not be more than 6. And the minimum distance between two adjacent discs should be 24 from center to center. The disc should never be relocated after being placed as some of the drug diffuse almost instantaneously. Within the 15 minutes after the discs are placed, the MHA plates are placed inside the aerobic incubator. The temperature of the incubator should be 35 degrees Celsius plus minus 2 degrees Celsius.